Good afternoon, everyone. My name is Kari Luther Rosbeck, and I am delighted to be co-chairing today's pre-conference workshop with my dear friend, Dr. Rodney Samaco. I'm representing the patient advocacy community and in my day job serve as president and CEO of the TSC Alliance. Dr. Samaco is a legend among our rare disease community. If you've ever been on a Zoom with him, he loves to proudly display all the swag from the rare disease patient organizations for which he proudly partners. Thank you all for taking the time to join us. This workshop is appropriately titled From Discovery to Approval, Understanding the Pathway to Accelerate Therapeutic Development. Before we get kicked off, I wanted to review just a few housekeeping notes. First, you'll notice a number of boxed sections to the right and left of the video attendee view, all of which can be minimized or expanded. The chat allows you to interact with each other and program staff, and we hope you use this opportunity to make some new connections. The questions box is located to the left of the screen in the attendee view, below the chat and polling. We encourage you to use the questions box to share your questions with the presenters. This is your time to find answers and insights to help you advance your work as a partner in research. If we are not able to address all your questions, we'll do our best to follow up afterwards. To the right of your video, you'll see captions and translations. To utilize either, please expand that session and select your language. Thank you to Horizon and Greenwich Biosciences for their support of these important tools. Additionally, you are welcome to utilize the notes section to take notes in the player. They will automatically be emailed to you when the session concludes. If you have any questions about the technology or the program, please look for Request Support button in each session or the live support chat in the bottom right corner on all session pages. We want to encourage you all to participate today. Take advantage of hearing from leaders and experts, and most importantly, ask lots of questions. When I first started in my role as CEO, there was so much to take in terms of developing a drug discovery pipeline. Like many of you, I struggled with exactly where to start to make a difference. I sought out every single rare disease organization that had developed a drug development pipeline that had come before the TSC Alliance. I went to every single conference, and eventually we built research tools slowly over time that we could sustain. Today, technology and science are advancing at a record pace, and there are so many opportunities for you to advance research in your disease states. Today's workshop is about walking you through some basics in drug discovery in a safe, unjudging, nurturing environment. We have a dynamic group of speakers that represent the spectrum of patient organizations and companies that focus on rare diseases. We also will distribute some helpful materials afterwards. And of course, we have thoughtfully created a relevant and impactful program for you, and are really grateful you've chosen to spend this time with us. It is now my honor to formally introduce my co-chair, Dr. Rodney Samaco. Dr. Samaco is the assistant professor and investigator at the Department of Molecular and Human Genetics at Baylor College of Medicine. His research program focuses on optimizing the framework for preclinical readiness of rare disease models. His team evaluates the natural history of disease in genetically modified rodents to identify measures that may serve as surrogate endpoints. By fostering collaborations across the landscape of patient advocacy, academic, and industry groups, he strives to advance community-based participatory approaches in research and development of actionable therapies for rares. Dr. Samaco is moderating our first segment, The Building Blocks of Disease Understanding and Preclinical Research. Rodney, it's all yours.
Thank you, Kari, for that wonderful introduction and very kind of you to speak of me in that way. So I'm Rodney Samako. I will be moderating the first part of this session on a lot of the preclinical questions that we're engaged with as a community when we speak with our various stakeholder groups, whether they be researchers, patient advocates, et cetera. There are a lot of, I would say, questions that have emerged, especially in the context of advanced therapeutic modalities and development of them. So I would say that let's start first, though, with the journey of an individual, Donna Appel, who I will let her introduce herself prior to moving into this session, because Donna has a very interesting story and she could share with you her lived experience and her challenges and successes in building a foundation. Donna.
Hi, my name is Donna Appel, and I am so grateful to be presenting here today. I know that you can all kind of relate to this story, but I'm kind of here to set the stage. I represent Hermansky-Pudlak syndrome, and it's an autosomal recessive single gene disorder. Presently, we have 11 different gene types. It's characterized by albinism, legal blindness. There's a bleeding disorder with all gene types caused by a platelet dysfunction. In about 20% of our folks, they develop a Crohn's-like colitis. More seriously, 100% of the time in HPS 1, 2, and 4, they develop a fatal pulmonary fibrosis. And without a lung transplant, they succumb in their early adult years. My daughter was born with albinism, with what we thought was albinism. And then she hemorrhaged to shock in her crib at 2 years old from the bowel disease. She bled so badly, she needed 36 units of platelets, 6 units of blood. She sustained a traumatic brain injury. And if we had known more about HPS, that would have never happened. After 3 months of hospitalization, I knew I had to find the experts. I loved her so much. I had to find the research. I had to find the cure. I found nothing. I started the HPS network and began this crazy journey of trying to cure disease and find research. It was beyond daunting. We came to find out that Ashley had the fatal type of HPS, and now we're racing a clock. So besides working full-time, being the caregiver to my daughter, the mom, to my son, the wife, I now had to learn research, genetics, pharmacology, and politics. And mostly I had to figure out how to go about marketing a disease, especially one with such a stupid name. My organization challenges didn't help when I established it. Our people are visually impaired. We have a founder's effect in Puerto Rico, so many of our families are Spanish speaking. We do a lot of work on the island of Puerto Rico, and connectivity can really be a big barrier to engagement. I send out great information and education, but if people can't connect, that's a bigger issue. And that island, they have hurricanes and earthquakes, sandstorms, and things become insurmountable at times. Our biggest hurdle, of course, is our tiny budget, just like everyone else feels. So where do I begin? Like, how do I go? What direction do I go in? So first you got to learn, like Kari was saying, you just have to learn and keep your eyes open. I remember in the very beginning calling for help. I actually called the Cystic Fibrosis Foundation, and they were so kind to me because they were the first people that taught me the difference between bench research and clinical research. Picking up the phone makes such a difference. Not knowing how do I know where the cure is going to come from? So I just started looking at the two types of researches and made a list. And I began growing them simultaneously. I wanted to investigate stem cells and organoids and gene therapies and things that we're going to learn in this panel discussion this morning. But I made a list. It was my to-do list to find bench researchers. And then I wanted to make clinical experts as well. So it was like growing our research one seed at a time. I just kind of worked it out and thought of the acronym DOC. I wanted to look for docs. So it became discovery, opportunities, and commitment. The first thing I needed to do was discover what's out there. I needed to expose the cause and do outreach. And then making those contacts with umbrella organizations like our hosts, the Global Genes and the Orphan Disease Center. These people are amazing. And finding my metabolic pathway partners, who's next to us in that gene. That I set our Google alerts and our journal alerts so that every morning I would get an influx of things in my inbox, and then I would read them and call those authors. Also finding out where the researchers live. We needed pulmonary research, so I worked very closely with the American Thoracic Society. We have a booth at their conference, and I'm on the Public Advisory Roundtable and have been on their board of directors. I even just cold call researchers. My biggest victory was a cold call to the NIH, and believe it or not, I got Dr. William Gall. And to decide about taking on HPS 2 decades ago, he asked us to collect 24-hour urine buckets from any families we knew with HPS. We had to do this in the winter because they had to be refrigerated. So we put them on our deck in the snow to keep them cold. It was life-changing for our organization and the beginning of our clinical research journey. We also took lots of road trips. My kids always thought that they were going on summer vacations in cool places like Niagara Falls, but it was really Roswell Park Cancer Institute to meet the PhDs that had the mice labs. We went to Maine and it was Jackson Labs. It was a great discovery for the kids, but it was really just moving science. After we discovered who's out there, we knew that I needed to provide opportunities. So we host a science meeting called the Meeting of the Minds. It started with 4 or 5 researchers, and now we're up to 50. Annually, they get together and talk in a space that's very collegial, but they can only present their work if it's unpublished data. Each year, I invite a guest speaker to come. We think of a subject matter like that we want to learn about, like an organoid, for instance, and we look around the country and try to invite best in class to come to the conference and be our rock star. They don't have to know anything about HPS, but the trick is I ask them to go last in the speaking agenda so that they listen to all the researchers that discuss HPS, and maybe by the end of the day, they apply some of it to their particular subject matter. And of course, involving always young investigators with poster sessions and the like. We combine all of that with our family conference because we want our researchers engaged with our people. We actually do an English version and a Spanish version separately rather than a translated conference. We do them separate. We really want to ask our researchers to meet our families, so we always break bread together. I guess, sadly, it's what I got, but I'm not ashamed of our small budget. We've supplied mouse food, and we've covered annual bills for electron microscopy repairs. We've given office equipment, and now we're grown to provide salary for postdoc. We also are creating experts by providing CME classes. We help with clinical trials. We're recruiting for our symptom scale study. And we really try to bring our researchers along by giving them our biospecimens. It's our culture. We have a program called We're Drawn Together. So we invite our researchers to bring the science to us rather than us going to the science. They run IRB-approved clinical projects. One year we had 5 of these going on at the same time. We had to have space for obtaining consents, which all had to be in Spanish and large print. And we had stations with my friends as phlebotomists, and we ended up drawing 98 specimens of blood and 4 stool specimens, and we accomplished it all in 1 hour and 55 minutes. So after providing these opportunities, now we want commitment. We need this research to be committed to our cause. We continually have ruthless groveling. And here's a picture of my daughter, that we sent to our researcher who's studying the CRISPR-Cas9 research. But we want to help them grow their peer group in our research. And we want to show our commitment by showing the growth of our cohort. We want to be able to establish a research-ready organization. So our job is to prepare our patients and our families. So we created the individual research plans. We identified the barriers for each and every member of our organization and figure out what those barriers are. If it's fear, we find them a buddy. If it's money and they can't afford a day off from work, we provide them support. If it's transportation, we get them to research. So we try to look at the barriers and try to mitigate it. We also make sure we have open communication with our researchers and actually help them communicate with each other. So we're creating a forum. And for our new investigators, we're always sure to tell them that we're there to help. We could provide speakers or send pictures or slides. We could write letters for supports of their grant. We make sure they know that we are willing to help. In summary, I just kind of think if you got to find your docs, I think of Doc. Discover who they are, offer opportunities, and inspire their commitments. I know it sounds kind of like a little Pollyanna-ish, but we got to start by just beginning, and this is pretty much gets built over time. And I am really excited to hand this panel back to our amazing scientists that are going to actually, I'm going to learn a lot, and I'm so glad to be here. I want to thank you so much for listening. I want to thank our organizers, and thank you so much for your time.
Thank you, Donna, for that wonderful background and the entire story. It's just amazing. And I think a lot of those vignettes that you shared with us today will be very helpful and encouraging for people who are part of the conversation. So now I would like to move forward with our scientists who are going to provide some of that background knowledge and domain expertise in terms of understanding the landscape, the lexicon, having the literacy to understand some of these basic blocks of disease understanding and basic research. Sometimes when people are talking about CRISPR or organoids or mouse model, conditional knock-in, knockout, et cetera, could be very confusing. And in addition, once you understand what those topics might mean to you or your, I would say, group, perhaps it's not entirely clear which path you should pursue. So today, we have 3 individuals who will speak about the building blocks, preclinical development pathways and inflection points, and the business of what we like to call the process. And we have James Doyle from Modelis, Ralph Schmidt from the Perelman School of Medicine, GTP program, University of Pennsylvania, and Yale Weiss from Ultragenics. And so we'll start with this topic. What is the intent? What is your motivation? James, please.
Thanks, Rodney, for the introduction. So everybody, it's great to be here. It's always fun being part of the Global Genes and this rare disease community, which is very open to collaboration. It's something which I really love about this field, and which is very different from the rest of most other scientific fields or disease areas. So it's really a pleasure being part of this. It's going to be great kicking this off with some of the research tools which are available to you. So a little bit on my background very, very quickly. So I am a scientist by training. I have a PhD in experimental medicine with a focus in molecular genetics. And I took a non-traditional route, going the biotech and entrepreneurial route and founding Modelis. back in 2018. So to kick this off, we'll begin discussing some of these cell-based models, or these in vitro models, which are available for a variety of research uses. And we'll use this time to go through some of these models quickly and go a bit deeper into them later on in the discussion. So among these cell-based models, the first ones that we'll touch on are cell lines. So these can be obtained from human or animal tissues, such as fibroblasts that are usually obtained through a skin punch. Then we have iPSCs that are stem cells that can be differentiated or turned into basically any cell or tissue type. And these can be generated from skin or blood cells and are patient-specific. But in the absence of any patient-specific lines, we do have these CRISPR-edited cell lines, which are available. And these are using CRISPR or genetic engineering techniques to mimic very specific patient mutations or very specific genetic variants in the cells where you don't have any patient-specific cell lines available. Thank you. So while the models on the previous slide are what we call two-dimensional models, there have been recent advancements in the generation of what we call organoids, which are these tiny three-dimensional structures that can be grown using iPSCs, or the induced pluripotent stem cells, to study specific tissues or organs in a disease state. And these can be made into brains, livers, pancreas, even muscle cells. And so while in vitro means in the glass and typically refers to cells in a dish, in vivo here means in living and refers to simply animal studies, or more generally studies in live multicellular organisms. This is really a topic which I'm very passionate about since I use these very regularly. We at Modellus use these for drug discovery. And so while mice are typically the most common, most well-known of these animals, there are many others that can be used to answer a wide variety of research questions, from the very small model organisms, such as fish, zebrafish, frogs, or xenopus, flies like Drosophila, and worms, C. elegans, and it goes all the way to the larger mammals like pigs, dogs, and other non-human primates. And each of these organisms have their own strengths and limitations, and while each can be used to answer specific questions, having multiple ones in your tool belt can really help you answer the widest range of research problems. And as for the types of therapies that are typically available to you, the most traditional ones are these, or we'll quickly go over these traditional categories, traditional modalities, which are small molecules. So think of your typical pills like Advil, which often can be taken orally. Then we have biologics, or protein-based therapies, which are usually engineered full-length proteins such as antibodies or enzyme replacement therapies, all the way to peptides that are short proteins like insulin, which is probably the most common of these, or the most well-known of these. And while we did name these traditional therapies, it's not because they're out of date, it's just because they have a very long and historical use as therapeutic classes. And finally, one more approach, which we'll just quickly touch on, one more tool in your tool belt which can be used, which is leveraging computational approaches to identify or repurpose drugs. And so aside from all the biological, so the in vitro and the in vivo approaches, there's another aspect which we just quickly wanted to touch on, which are these computational-based approaches, also known as in silico tools. And these are most often AI-driven, that look to predict molecules by leveraging data such as molecular targets to design very effective or very specific drugs to very specific target proteins. And that can be incorporated or aligned with any of the other strategies we had discussed before, any of the other tools we had discussed before, and also kind of going back to an important point, each of these can help you answer specific questions, and one of these is usually not the be-all, end-all, and the broadest range of tools in your toolkit can really help you answer the widest range of questions.
Thank you, James, for that great background introduction. For everyone in the audience, please, if you have any questions and or comments, please put them in the chat box. And I would also like to inform you that these slides will be available in the later as part of content, as part of the workshop. So if there's anything that you missed, we have the recording plus the slides. So moving on to Ralph. Ralph will discuss some of the therapeutic modalities with respect to the genetic intervention.
Needs to unshare. Okay. Yeah, just a moment. Just hold on a second. Hello, my name is Ralph Schmidt. I'm a researcher at the University of Pennsylvania. I work here at the gene therapy program, and my group is primarily interested in rare neurological diseases. In the following slides, I will give you a few talking points on gene therapy and viral-based approaches. Which is directly complementing what you just heard about using small molecules to try to cure diseases. So why are we interested in anything with the word gene in it? Well, one of the reasons is that the vast majority of rare diseases, about 80%, are genetic in origin. So we really have an opportunity here once the genetic defect has identified trying to directly alter the genetic disposition of the patient, where small molecules typically just trying to alleviate symptoms and kind of curing around the genetic defect. So how do genes work? And I just want to take a step back that you all understand the process. Normally we start with the gene is encoded by DNA. That gives rise to an intermediate form, which we call RNA, and then eventually we end up with a protein. The protein travels to where it has to be in the cell and fulfills its function. So now if there's a mutation in DNA, a pathogenic mutation, that precludes the DNA from turning into RNA, and then there's also no protein, or the protein is so short-lived that there's essentially none there. And that is true for a lot of the genetically encoded diseases. So now the opportunity then basically is to trying to repair this right on the genetic level. So the one thing what you can do is gene therapy. So you basically add the gene, you add an additional gene, the same gene which is already there with the mutation, but you just add the fully functional one. You add DNA for that. Or you use something which is called gene editing or CRISPR, as you heard before, which are tools to basically revert this mutation and turn the mutation into the normal genetic code. Both methodologies then basically would allow to produce RNA, then we get protein, and so then the cell can resume its normal function. So now, gene therapy and gene editing, both methodologies consist of really two components. So the one thing is our delivery vehicle, and that could be Something we see all the time, which has been established in research already. They like the FedEx vehicles or planes, you know, all about. But then this could be also something which is emerging or new. FedEx analogy would be a drone or a robot. And so this one, and I will expand on that a little bit more. And following slide, the delivery vehicle is typically virally derived, or it is some chemical mechanism how to get this piece of DNA or the gene editing machinery actually into the organism. And then the other important component, of course, is the payload. And so the payload now, that those are the packages being transported by the FedEx trucks, they can be of various shapes and sizes. So what that allows to do is really you can do a direct modification, a gene replacement. So one of those boxes would have the gene in, gets put in the body, and it ends up in every single cell and is starting to make the protein which was lost. The indirect approach would be that this package includes instructions how to regulate gene activation. So that could help to either switching a gene on, if that is helpful for the disease. If, for example, in some diseases, you still have a normal copy of the gene left, so you could switch that on to overcome the disease symptoms. Or in other cases, it might be that there is a mutant expressing, and by simply getting rid of that mutant gene, mutant protein, you can improve the disease symptoms. And you can deliver those regulators, and they're highly specific regulators. You can just target the gene of interest. And then the other indirect method, of course, would be the editing. You can edit on both levels. You can either edit the DNA itself, that's the human genetic code, that's kind of a one-time and done thing, so that's a permanent change to the human genome. Or you can edit the RNA, that's intermediate before the protein is made, that is not permanent. So you're not touching the genetic code, you're just basically repairing what comes out of the genetic code. So why is anything with gene therapy, gene editing, really, why might that be game changing, and why there is so much buzz about it? Well, what we think that those technologies can really provide a single and durable administration, one-time administration. And I think one of a good example is SMA gene therapy, so Zolgensma, which was approved a few years ago. So right now, some of those patients are in 6 years post gene therapy. So they were giving gene therapy to repair the genetic defect, and then the condition kept improving, and the children are still very mobile and have overcome a lot of the disease symptoms 6 years after a one-time treatment. Of course, time will tell how long really this single durable administration will hold, if it's really decades, or if, let's say, after 10, 20 years, you need to do some kind of re-administration. So now let's talk about the different modalities, how you can actually administer your gene therapy or gene editing. The one option is viral gene therapy. So what you typically would be using is just the shells of a virus, also called capsids. And I have a picture, computer animated picture on the left side here, which can provide really efficient delivery. And I know, looking at that, you probably say, oh, well, this just looks like COVID, which is true because it is a virus. But in this case, we would only use the outside, not any of the payload, which is inside that virus. Because given for COVID, for example, it is really what's inside that virus what makes people sick. So the technology is throw all of this away, and basically we just rely on delivery vehicle, which is the outside. So for human therapeutics, we mostly rely on lentivirus or adeno-associated virus or AAV. Lentivirus, it integrates permanently in the genome, so it just adds itself basically to the human genetic code. So it's really used a lot for ex vivo manipulations, where you would take patient cells out, you modify them, And then you do a lot of quality control steps before you re-implant into humans to make sure that everything is okay, since it's a permanent integration. AAV and on the other side is the AAV genome, the payload, persists separately from the human genome in the cell. So you're basically just adding an additional piece of DNA, which is also functional inside the cell. And that's why this is really the workhorse now for in vivo applications, where you would directly inject the AAV loaded with a gene therapy or with the gene editing tools into the patient. Another modality is adenovirus. You might heard about that. That's the basis really for the J&J vaccine delivering COVID spike genes. It is very immunogenic, so that's why it's really good if you want to cause an immune response and train the immune system. But it is less optimal if you want to cure disease, which has nothing to do with the immune system. The non-viral or chemical gene therapies, which can be used, those are for the most part liquid nanoparticles or LNPs. That is really an emerging technology, but I think it really leapfrogged into the public view, and we learned a lot about it because that is the basis of the Pfizer Moderna COVID vaccines. And so LNPs are basically another way to deliver genetic information, either DNA or RNA, to the patient. Then the question is, of course, well, replace a broken gene by a gene therapy or just fix it by gene editing, and that really depends on the context. What you need to achieve, if you really need to replace something, if you need to down regulate your gene. So what your target organs are, if you have one target organ, if you have multiple target organs, your patient population, if there's one prevalent mutation, and basically targeting that, you would help to cure a large set of patients, or if it's really a lot of different mutations, and so rather replacing the gene. And of course, I mean, it's what is the safer way? What is the safer way in the end to overcome a genetic disease? Okay, so real quick, some routes of administration. So you can either apply AAV technology intravenously, just injecting into the bloodstream. You can directly inject it for CNS. It would be in the brain for the most part. All the other organs are easier reachable for IV. Or you can go for CNS indications. You can go through the CSF, the cerebral spinal fluid, which is either intrathecally, where it's administered by a needle in the spinal canal, or by injection directly in what's called the cisterna magna, which is closer to the brain, as a fluid-filled cavity. Those three modalities on the right are really the good choices right now for any CNS indications, because unfortunately, with IV, you can reach about any organ except for the brain, because there's something called the blood-brain barrier, which really precludes anything from seeping out of the blood and entering the brain tissue in an efficient way. So people have been trying to address this for a while now, trying to find AAV, specialized engineered AAV, which could do just that, because AAVs are normally kept hindered by the blood-brain barrier to cross over. It has worked really well in mice. So if you inject mice with green fluorescence protein DNA encapsulated in AAV, the brain lights up very green. Unfortunately, researchers still working on translating this kind of spectacular approach into that it works also in primates. So that's why really, I mean, delivering to the spinal cord, into the CSF, it's very efficient. Neuromuscular disorders can be addressed that way. Direct injections into the brain or any other nerve tissue, really it has to be a specific cell population. It's highly local, for example, like for Parkinson's disease. Global brain delivery of AAV gene therapy to the brain is at the current point somewhat inefficient. It works really best if you have something which is called a bystander effect, where the cells which receive your gene therapy then can secrete the protein after it's made, and so repairing all the surrounding cells as well. But that's obviously a highly specialized situation where you're dealing with secreted proteins. So this was my part talking about AAV gene therapy for the most part, and now I'd like to turn it back to Rodney for the next section.
Thank you, Ralph. That was a great overview of the different modalities, and we already have some questions from the audience about this, and we'll touch upon it during the panel discussion, especially with respect to antisense oligonucleotide. So next, we have Yael Weiss, who will talk about the business of science. Should this matter and why? Yael.
Thank you, Rodney, and thank you for Global Genes for putting this workshop together. I hope that this presentation, this whole workshop will be helpful for foundations that are starting out and really have to make a lot of decisions on which paths to take, and also foundations that are already underway and have some idea, but maybe we will be opening other horizons for them or enabling some decision making as well. I won't spend too much time on the business of science. This is what I do on a daily basis. I'm part of the business development team at Ultragenics, which is a company focused on rare genetic diseases. I just wanted to focus on two topics that come up a lot when we speak with patients and foundations. And the first, which is actually really important, is how should you be providing funding to an academic? Should it be a grant or a sponsored research agreement? And did you even know that there's a difference between the two of them? The grant mechanism is usually very simple. It takes less time, which is always more attractive. It's usually cheaper because you don't have to pay for a lot of the overhead costs that universities usually charge when we put in place a sponsored research agreement. However, it comes with less strings attached, which is sometimes advantageous, but many times not, because you don't have any rights to anything that's generated out of the research that was funded. And in some cases, you may not want it or not need it, but you need to be aware of the fact that if any inventions come out of the work that you funded or any important information is generated, you don't necessarily have rights to it and you can't really do much with it. So in some cases, it's not an issue at all. You're just happy to get someone working on your disease and understanding the biology better and maybe publish so that it's available to others. But in cases where there is a therapeutic option and you want to control the fate of this therapeutic, you may want to look at the sponsored research agreement route. It's more expensive, as I said. You're paying for more infrastructure in the university, but you can get rights to any patent or intellectual property that was derived from the research and access to the data. So it takes longer, it's more expensive, but you get a lot more in return. So definitely when you are starting discussions with an academic, make sure that you understand what the end result of this research would be and what you would like to do with it when it ends. So that's just something that we've heard over and over from foundations that provided grants and then did not have any rights to the data, and the physicians either license the IP to somebody, or the academics license the IP to someone who didn't advance the science and just lost the rights to the foundations lost the rights to it. And then 2 words on whether patents are important. So if you want to work with a company, yes, a patent is important and highly recommended that the academic or the foundation, if the invention comes from the foundation, that you file a patent. Costs money, I know, costs money to maintain it, but highly worthwhile because it gives you a strong position when you're talking to pharma, biotech, or other partners that might want to continue development of your inventions and take them into the clinic. There is a regulatory pathway called orphan exclusivity that sometimes can bypass the patent because the regulators will give you this exclusivity in the marketplace based on the fact that you are there first with an invention that no one came up with before. But the combination of having a patent and orphan exclusivity is usually the strongest for any company that wants to market a drug for a specific indication. So just the highlights, and obviously the discussions are much longer. I actually really like the definition of building a toolbox as a foundation when you're starting out. We see a lot of foundations that have that come to us, and they have the toolbox on the left-hand side. They collected things from all over the place, not in an orderly manner, and don't really know what's in their toolbox. On the other hand, the right-hand side is a very nice and orderly toolbox. I don't have that one personally, but I'm sure people do. And it allows you to build your toolbox more systematically. If we can go to the next slide, Rodney. So what is a systematic approach to creating this toolbox or inventory list of what's inside your toolbox? So when you're starting out as a foundation, first of all, you need to understand what's already available. I mean, are there cells? Are there mice? Are there organoids? What has already been done, and where has it been done? And do you have access to it? Now, once you have that list, you start identifying the gaps in it. So if there are cells, but there isn't an animal model, then this is something you probably would like to focus on. If there is an animal model, but it doesn't necessarily, it's not the best animal model for a gene therapy, then maybe you want to consult with Ralph or somebody who's a gene therapy expert on maybe you need a different mouse. Who will make it? Will it be an academic lab? Advantages, disadvantages of working with academia versus a company, price, timeline, publications, etc. What would be the price? How long will it take? How do you, and as I alluded to previously, how much control do you have over the process, oversight, milestones, payments, etc., and who owns the results at the end? So I don't know that we need to go through all the lists here and every point. For each tool that my colleagues and my fellow panelists presented previously, there's obviously a decision to be made, whether it's needed, whether it's relevant, and how much to invest in it. Obviously, if you're just starting out and you're not a very wealthy foundation, you need to focus your efforts and decide what the most important things are that you need to start your journey and get research going and get people interested. If you have abundance of resources, you will probably try to do more. But there are things that take longer. Mice take longer than cells. A natural history study takes longer than a registry. So you also need to put things on a timeline and understand where you are now and where you want to be in a year or 2 or 3, and then start things that take longer, right, when you're kicking off things versus waiting, and then it will take a lot longer to get the information of the day that you need. We always recommend to make cells. We always recommend to have an animal model, but again, it's a matter of resources and timing. You go to Rodney and you ask him, which animal model should I make? Or what's the best animal model for my disease, my gene? Do I need more than one model? Do I need a knockout or an inducible model? And Rodney will explain to you exactly what each is. Does the model have to be 100% identical to what the clinical presentation is? Is it even possible? And what happens if it's not possible? And then biomarkers are important, especially starting preparing your biomarkers towards clinical development. In diseases that the endpoints take a long time to follow, a neurodegenerative disease, for example, or a fibrotic liver disease, you want to see if the drug is working sooner better than later. You can't wait years and years to see a difference. So you want to be able to follow specific biomarkers, whether they're blood biomarkers or an imaging biomarker or an EEG biomarker, depending on your disease. You want to be able to establish this as a good biomarker for your disease so that it could be added into the clinical trials. And this will be done way before you go into the clinic. And another thing that we recommend, and I think this is maybe the strength of the foundations and the communities, is really know your community and know what a company like Ultragenyx or any other company needs to know about your community in order to help you develop a therapy. How many patients have been identified? What are the sexes, the age distribution at diagnosis? How many different mutations are represented? Is it, as Ralph said, if it's only one very prevalent mutation in most of the community, then there might be a gene editing approach. But if each patient has their own unique mutation that all leads to the same clinical presentation, then the therapeutic approach will be different. What are the most common clinical symptoms? And as a company, at least at Ultragenyx, when we start thinking about running a clinical trial, the first thing that we will do is survey the community and try to understand what each family and each patient actually feel are the most important things that they would like to see corrected or changed in their disease, so that there is a correlation between what we think is measurable to what you think is significant. Develop a registry if possible, and this is usually easier, and there are many companies these days that can help you develop registries. And more complicated and more time consuming and more resource consuming is the natural history study. And the difference between them is the registry usually captures only single time points, while a natural history study looks at a disease over time. And the reason that we as a company and other companies as well prefer a natural history study is that in many cases, we can actually use the natural history study as a comparator group in a clinical study. And this saves time, saves resources, and also allows to run a smaller trial with less patients, which will be faster. I think we are done.
We are done. Thank you so much to our panelists for sharing their knowledge and domain expertise and addressing some of the basic building blocks when it comes to these terms in terms of the basic science, but also the preclinical modalities. And now, time for discussion. And for the panel, so we are all now live, and please unmute yourselves. I do have one question, and I think it is to prompt or at least prime the discussion for additional Q&A questions from the audience. But I would say if I was sitting here and I was a father of a child, for example, with a rare genetic disorder, just finished the diagnostic odyssey, and now this is like blowing me away, right? Where do I even begin? I understand biology, let's just say, I understand that. I did study molecular and human genetics, so that's a plus there. But let me ask, how do I begin a foundation when it's just me alone? Let me ask the question of when I see fish, zebrafish, for example, flies, worms, mice, non-human primates, where do I start? Let's say if I had unlimited resources and wealth, great, but I don't. So how do I even fundraise? How do I make actionable what I have tangible in front of me with known quantities? So let's start there. And I will either call on you or you could start off, whoever's the most eager and enthusiastic. Okay. Yael, since you just spoke, let me start with you and we'll work backwards here.
I don't know that I can answer all your questions, Rodney.
Well,
I think
exactly. And
that
that I think that's really valuable, right? In terms of we cannot as a community be something for everybody, but as a network, we can meet those needs, right? I think that is probably the number one the solution. And Donna, maybe you can
I,
speak to that.
I think that going to Kari and Donna is a really good place to start because they've gone through it. They have a lot of experience on starting from the bottom and going up and expanding their networks, expanding their knowledge. And while not one size fits all, right? It's so different for different types of diseases, different genes, different mutations. The process at the end of the day is very similar because you need to have a critical mass of patients that you could collect evidence from. You need to have a community that you can collect information from, and you need to build a scientific network of basic researchers and clinicians who understand the disease and want to work on it. And I think Donna's story exemplified it perfectly. The path that she went through is probably the path that almost everyone will go through. I don't think there's any fast forward button here, unfortunately.
And I think it's unfortunate that you have to do all of it at the same time. And one of the things, so I love Yale's discussion. I mean, I want to capture, I can't wait to look at it again and write down every single thing that you said to do. So we're blessed that right now, if Rodney, God forbid, and that it would never happen, that you would need to start a disease group for something. We have certain lists now. There's, don't reinvent the wheel. You need to grab another group that's a bit ahead of you. My organization is super rare, super small budget, and Kari at the TSC Alliance, they're my best friends. Like, I ask these organizations for help. But you kind of have to be able to do many things at one time, building this road. But I think the biggest thing is to understand who we are. We need to own who we are, and every organization's got something different. So sometimes you might be, a parent that happens to be a researcher. So you're going to go this as a big science thing. You might be, I'm a mother and a nurse. I'm a little more nurturing for my people. So every organization is going to have its different flavor, but you need to begin knowing that what our job is in this is that we are not the scientists. We're not the pharmaceutical company. We're probably not going to find the compound or do the gene therapy. But what our job is to collect a really clear, defined cohort of people so that we can feed you guys everything you need. And that's our job.
And I do want to say that the people like you, Donna and Kari, you already took an extra step that a lot of patients or parents in your community didn't. So you are one step ahead of your whole community, which is not an easy task, right? You're taking a community on your shoulders, but you also, because you did it, you have the ability, the personality, and the strength to do it. So you have to be very aware of the fact that you are the one that is going to be able to execute this.
Thank you. That becomes a little frightening too, as well. It's like I can barely raise my children and be the caregiver and then run the organization. And I think the point of having to know all of this, you guys can only imagine how overwhelming it is for our, for families to take on science. But you're so wonderful in helping us adjust a discussion and course like this, where we get a list. Do this, do this, do this. It's like opening up a cookbook and looking at a menu. It's wonderful to be able to have those resources and just follow it. Don't reinvent the wheel, just follow along.
And it's okay if you miss an ingredient, and it's okay if the food is burnt sometimes. It's okay. I mean, just continue and try the recipe and it will work out fine.
You have to say that during lunch. I love it. So we're going to address some of the questions that were in the Q&A, as we only have 10 minutes. But if we don't get through all the questions, please join us, audience, in the breakout rooms later today. So a question, basic science question, and anyone can take this. Is there any limitation with respect to frameshift mutations? And I think the question here was in regards to the therapeutic modality, whether it be gene therapy, ASO, or drug repurposing.
I can speak to that. So it really depends on what the effect of your frame shift is. So if the frame shift is stop, no RNA, no protein, then it's clearly cut. So you can either replace your gene therapy, just replace, or you frame shift mutation is, in most cases, it's probably a single nucleotide. You can just try to repair that. If you, however, if you get stuck with a truncated protein or with something else, it might be more complicated. And you actually have to study. Sometimes there might be problems if you have a truncated residual protein. You're putting the wild type gene back, you get the functional protein, how they would interact with each other. So that's really on a case by case basis. You would have to look what the biology is.
Okay. There's an upvote on a question here, and this is related to sponsored research agreements. Aside from financial benefit, are there any reasons to opt for a sponsored research agreement? For example, what additional control do we get over the future of research in therapeutics?
So when you put in place a sponsored research agreement, you can actually put in an option to license any intellectual property that comes out of the sponsored research, which in a grant you do not have access to. And you can also put in clauses that give you rights to information, looking at publications before they're published, having milestones that the academics need to meet. And if they don't meet them, you can theoretically stop paying. So it just gives you a lot more hands-on, I don't want to say control because no one wants to be controlled, but insights into what's going on with the work that you funded.
Great. Another question, I know some of these are jumping around, and that's okay as they're being upvoted. What are the best computer tools for developing a therapy? What would be a good starter set of software?
Oof, that's a really charged question. I don't think there's a one answer to that. There's many different approaches out there. There's many different companies, biotechs, academic groups who have different solutions to try and identify therapies based off various algorithms. I don't think that there's really a one size fits all answer to that. But I would just begin researching some of the options out there, looking at trying to identify what are you trying to do, or are you looking to identify a small molecule to target a specific receptor? If so, what are some of the tools around that? And how could you design a molecule specific to that receptor? But yeah, it would really have to begin with looking at what's out there in terms of solutions and which one is best applied to your specific problem.
Great, thank you. Another question about gene therapy. So is there any concern or indication that gene therapy will not sustain over time?
We don't know yet. So I think it holds the promise that the effect will really, really long lasting. But if you're thinking about a pediatric disease, so you're talking about that you're trying to make the effect last for like 80 years or something like that. And so nobody can predict. Nobody can predict at this time. But compared to other modalities, it's definitely longer. So if you're thinking about antisense oligonucleotides or ASOs, they require frequent re-administration. So about 2 to 3 times a year. Lifelong, so that there's definitely a difference with other modalities in terms of how often you would have to administer.
That's great. So I know we're hitting to the mark of break, but maybe we could go a little bit past the time just to get some of the questions addressed. But you do raise an important point, Ralph, related to this question that is still unknown at this time. Perhaps what are your thoughts, and maybe Yael, James, Donna, and others who may be here about polytherapy? So when it comes to genetic-based therapy plus supplementing or complementing that with the different modalities, such as a drug that may improve or been shown to improve quality of life.
It's a great question, Rodney. I think this will change over time as gene therapy develops, and as Ralph said, more will be known on how long it lasts, and do you need to redose, and can you redose, and if you can, do you need to augment with another therapy? Sometimes gene therapy will address major organ symptoms and not everything, so you will still need to augment with something that treats the organ that's not affected by or improved by gene therapy. And a lot of patients will come into a gene therapy study or a gene therapy therapeutic already being treated with many medications. So you'll have to slowly start tapering down things to see what you need to stay on and what you can let go of. I think as more and more gene therapy products go into the market, we'll know more and more.
That's great. I think there's some really interesting, intriguing questions here in the chat. This is for Yael and also for others, if you want to comment about the perception that biotech and pharmas are not interested in finding and investing in therapies for rare disease because they may not be money makers. Populations may be too small. And for something like COVID-19, which impacted the world, and that was given as an example, it's a big money maker for companies. Is this true? Is this sentiment true? What are your thoughts? Can you share some insight in terms of the corporate investing in rare disease? Do they really care?
Well, I don't know if I'm the right person to ask or the wrong person to ask, because I work at a company that's focused only on ultra rare genetic diseases. So obviously we care. We are limited by limited resources. There's only so much we can do, but we try to help everyone that we can, either by doing the boot camp or speaking at workshops like this. I think there's a set of companies that see the value in rare diseases and understand that many times you learn a lot about specific pathways in the body and about bigger diseases through understanding a rare disease, but not everyone, unfortunately. I used to work at Merck, and I don't think Merck would ever be interested in rare genetic diseases. On the other hand, Pfizer developed a vaccine and has a rare disease unit. So it's hard to generalize.
I would like to add, I think part of the problem is also just the number of rare diseases. So my slide had suggested there's at least 7,000 known rare diseases right now. So it's completely impossible that somebody is working on every single disease at any given time. So how it normally comes to place, it's really once there is a patient group, a vocal patient group, like Donna, for example, explained. I mean, if you reach out to researchers, help them to understand, or companies, maybe get some seed funding and so on, then it doesn't really matter, well, it somewhat will matter how many patients there are, but I mean, then it is less an issue if you have 50 or 50,000 patients here, because it is really to kickstart the research on it. That's the most important one. And then for some ultra-rare diseases, you might actually discover once you have a molecular test or a natural history study, there's a lot more patients than you thought.
Right. I do want to address one question about cognitive issues or for pediatric patients. Is there a delivery method that would be generally best?
For gene therapy? Or for
yeah,
general?
Right. In general, we'll take that in general, but also gene therapy. We could start with gene therapy.
I'll let Ralph take that one.
No. I mean, it is the same. It doesn't really matter. It doesn't really matter on the age. So there's the same limitations that intravenous can target most organs except for the brain. If you're trying to get in the brain, it becomes very inefficient. So that's kind of across the board. Of course, I mean, there's special considerations for still very early, very young patients. So like very young babies, there are special considerations. They, of course, because they're still developing in general. So you have to think about this more carefully, as if it's a teenager or even an adult.
But that's one of the things that we will ask in a survey. So if your child is very disabled and you can't administer an intranasal therapy, then we will try to make a subcutaneous formulation that the parent can inject the child. If the child can take a drug intranasally, then, and we can formulate it intranasally, then it has an advantage. So we try to understand what the physical limitations are that allow or prohibit from specific mode of administration and develop accordingly.
Right. And that leads to some of the questions that have been put in the box about surveys and registries. Do you think that they've run their course? They take up resources, a lot of them, and especially after a few years, can these resources perhaps be better used elsewhere?
From a foundation's perspective or from an industry perspective?
Let's start with a foundation's perspective and then an industry perspective.
I love the question, and I can't wait for you to answer it because it takes a lot of resources from the foundation end to run these registries. And, is it giving us cures?
So historically, actually, companies paid for natural history studies, which was great, right? Because it wasn't a burden on the foundations. But what happened then is that one company owned all the information and wouldn't share it with others. So that's what does the foundation think about that? That one company has all the data and not sharing it with others, and everyone either has to start their own or not have access to information. So the foundations having control over it is amazing. I don't think it needs to be funded by the foundations. Companies that are interested should participate in the costs, and then this will give them access to the outcomes. I still think these are important tools, especially for clinical trials, and especially for new diseases, new genes that we don't have much background and information on how they progress or not progress. And as I said before, if we want to use them as an arm, a comparator arm in a clinical trial, we need to have this information. But I don't think the foundations should pay for it.
In reality, though, it becomes very circular, because just like you were saying, that it's easy for companies to get interested in an organization, especially when they're organized and have registries, then it's hard for us to get the registries without the companies that are interested in us. So,
yeah, and I think the business model needs to be probably reconfigured. Talking to companies like Citizen or Allstripes or the companies that offer these services now, how can you put in place an arrangement that will allow you to kick it off, and then when you have something, pull in more resources?
I would like to add, I think foundations are absolutely critical in reaching out to patients, recruiting new patients, identifying new patients, linking up with the physicians. So I know it's a lot of work, but I mean, with a handful of patients doing a natural history study, it really doesn't give you the full picture. So I think there can be never enough patients, because you will always discover something or something you have not seen before. If you have a few hundred patients with the same mutation, suddenly a picture starts to emerge what the specific disease symptoms are in that subpopulation.
Yeah, I definitely agree with what's been said on the registry note. Like, it is a never evolving effort. It's never going to be done. It's a never evolving resource for the community to know who the patients are and where they are. But it does need the sustainability needs to be built into the model a little bit better. And whether it's getting sponsored, as Yael said, having companies sponsor these registries, but just something which is sustainable in the long run. Because yes, it is expensive to set up. It is expensive to maintain, especially. So there does need to be thought around building a sustainable long-term registry that's going to continue serving the evolving needs of the community.
Great, thank you, everybody. So we're out of time. Actually, we're quite over. We want to give everybody a break, but please know that we have the breakout rooms later today. This has been very enlightening and exciting and very pleasant to hear conversations so far. Up next, we have building a foundation for success in clinical research. So please come back and join us in about 5 minutes. Our next panel will be up. There are a number of questions that are still within the Q&A. We will find a way to address those either directly to you, or if you are at the breakout rooms later today in the office hours, we are happy to chat with you about organoids or foundations, and what you do when an academic researcher might leave, even specific questions to Ralph, STK11, and to James, something about organoids or expounding a little bit about organoids. Please join us later this afternoon. Thank you, everybody.
Thank you to Rodney and all of our amazing presenters from part one. They left very big shoes to fill. We're really excited to share with you part two of today's workshop, Building a Foundation for Success in Clinical Research. So now you have a foundational understanding of science and data sets, preclinical models, and translational research. We want to cover what it takes to move into clinical development and how patients can really be the drivers of this important process. What does it mean to be a good partner? How can we, as advocates or advocacy organizations, drive progress? I'm excited to be joined in this session by Vanessa Vogel-Farley of RareX and the Duke 15Q Alliance, and by Dr. Michael Grosso of Daiichi Sankyo. I thought that I might start by showing and sharing what the TSC Alliance has done to really build collaborative resources and tools to achieve and increase the interest in TSC drug development. Again, I want to emphasize we built our research platform over 35 years. And if we can just advance these slides to our research platform. You can see on the left that we have some tools that fit more into the previous conversation and are directed more towards hypothesis generation through research grants and drug testing in preclinical models, which are all meant to de-risk drug development in TSC. And on the right side of this graphic is where we're going to focus today and how we as advocates working in partnership with academia and industry can share our real world experiences on ad boards, through natural history databases or patient registries, by truly understanding the impact on quality of life through the collection of patient-reported outcomes. And in the case of the TSC Alliance, that's linked to our natural history database along with biosamples and how we can work towards building centers of excellence to drive clinical trial infrastructure, such as we have with our Clinical Research Consortium. All the tools at the TSC Alliance were built to push drug development from the left and pull it from the right. They're resources accessible to investigators, whether academia or industry, and definitely in partnership with our patient community. This is just one example of a research platform. We took time to build this by adding each element, as you can see, because the year that we put them into our research platform is listed, and in a way that we could sustain our program as a whole. It's scalable, meaning we can add more biosamples or more participants to our natural history database in a year where fundraising is going very well, or we can scale back in challenging times like during COVID-19. We built our program to be particularly flexible for that reason. The model I'm sharing talks about clinical trials and real-world experience. There are phases to clinical trial and development, and we'll include those in the overview of clinical terms in the resources that you receive after this workshop. But I want to start today's session by introducing our first speaker, Vanessa Vogel-Farley. Vanessa is the Research and Data Governance Platform Lead for RareX and also serves as the Executive Director of Duke 15Q Alliance. Previously, she worked at the University of Minnesota Center for Neurobehavioral Development and as the Clinical Research Coordinator for the Division of Developmental Medicine Laboratory of Cognitive Neuroscience at Boston Children's Hospital, collaborating with researchers at MIT and Harvard. She has over 15 years experience in data collection, nonprofit strategy, patient advocacy, and beyond. And I can tell you that this woman is a powerhouse. So Vanessa, I'm going to turn it over to you.
Thank you, Kari. I'm going to share my screen. And I hope you can see the proper screen. Give me a thumbs up if you can. Great. So as Kari introduced me in my background, I have now over 20 years of experience in collecting data, which is kind of crazy how quickly that adds up. And I am really proud as a person who's collected data over time, my 15 years of experiential data collection in the laboratories of cognitive neuroscience studying child development in that space, I really came to learn a lot about data collection, especially in rare disease populations, and how we needed to approach it maybe a little bit differently. And then during my 5 years of being the executive director of the G15Q Alliance, really being able to take some of that knowledge and those shared experiences and apply it to how we collect data at G15Q and in collaboration with Angelman syndrome. But today, I think what I really want to bring forward is the endeavor that is called RareX. And I serve as their research and data governance lead. And what the purpose of RareX is to really help bring forward some infrastructure and some shared learnings where oftentimes as rare disease patient groups, we're talked about, we stand in all of these places and say, we need to collect data. That's what we're told. You know, Dr. Woodcock from FDA, Dr. Mullins from FDA, CDC, all of the people go, the only case the best case scenario is collecting data to get your natural history study, that's the only way we're going to get to the end of towards therapeutics. And oftentimes they get off the stage and they don't tell you how. And what ends up coming that these patient groups and all of us patient groups end up having to become experts in all of the things that go into data collection, whether it be the infrastructure for governance, the infrastructure for actually the tech side of things, the infrastructure for data sharing, and all of these other areas where at the end of the day, a patient group, it's really, really tough to, as you're kind of bring your community together, as you're trying to figure out what your education looks like, they're trying to support all these families who are being newly diagnosed. This is an entire other realm of things that we need better support in making this a shared environment. So at the core of this, what RareX teaches to patients and its importance to progress is to accelerate disease understanding and the production and approval of therapies. First, by characterizing your disease. We talk about natural history studies. Kari talked a little bit about the Tuberous Sclerosis Alliance's pipeline where they have the natural history data. They have their real world evidence part. But how do we characterize the diagnosis, the clinical manifestations, the change over time, and all of those things? Oftentimes we talk about natural history studies. Everybody hears about those, and those are really, really paramount to collect. How does the disease present, and how does it change over time? Then figuring out and categorizing and organizing your data that's out there. Are there similar diseases to collect data in the space that you can really learn from? Oftentimes you feel like you're in your own bubble because we are all rare. But there's a lot more in common, especially when it comes to data collection and towards clinical trial development that we need to share. And so really going into that categorization and organization, saying, like, what can I learn from you? What needs to be changed and what can be similar? RareX really brings forward the standardization domain area, meaning that collecting standardized data is really important, especially in rare disease, because towards clinical trial development and in engaging with clinical organizations, like we'll hear about from Michael, it's really important that we are collecting data in the same way. Though oftentimes we do need offshoots. We need things that are specific to our genotype, and that comes a little bit later. But making sure the foundation of our disease characterization and our disease data collection is in that standardized way. This really helps you to identify the therapeutic targets. What's out there? Is it new drug discoveries? Is it drugs that are out there for repurposing? And also the design of your clinical trials. In rare disease especially, we become partners with biopharmas that are coming to the space because we're the experts in our disorders. We're the experts to say, this really needs to be an outcome. This is how you can identify it. This is how you can see over the change over time. And if we're collecting data at the beginning and in a way that's really, really useful, our biopharma partners can utilize that to really design a clinical trial that's going to hit the mark. We see too many times that these trials are designed in ways that it doesn't hit the mark. And if through data collection from the beginning, being very, very careful, it's going to help us accelerate all of these approvals of these clinical trials and then drug marketing and approvals. It helps also determine outcome measures. Oftentimes the outcome measures that are out there, because we're rare, might not be ones that are really beneficial to the community, or what might not be ones that hit the mark again. We are often talking about biomarkers. We often talk about quality of life. We often talk about, in my spaces, we talk a lot about seizures. We talk about epilepsy and seizure number. Are those the best outcome measures for the drug target or what they're really looking at? Or do we need to have some experimental things or secondary outcome measures that can be tried throughout the process? And have other experimental therapies made a difference? Or anything else that there's interventions that your community is going through that you could say, oh, this seems to be something that's changing the trajectory of the disorder, or this is not, but it's oftentimes used. Then engaging with your patient and support with our quality care. At the end of the day, data collection and the characterization of your disorder really means a lot only when you bring it back to your patients. It really needs to be brought back into your community. The data you're collecting, how you're using it, what characterization is coming forward, how biopharma is using it, and it can be utilized by biopharma. And additionally, any physicians or medical professionals or researchers who are able to use that data is really, really important too, because you're going to improve your patient and community engagement, and you're going to find your best practices and best treatments through data collection. This is kind of the soup to nuts of what it really means to be a great, setting up a great organizational data collection structure and engagement. Because at the end of the day, the key to rare disease clinical research is to ensure that data collection meets the needs of all stakeholders and ensures that it really is truly patient-centric. And in order to do that, education around effective data collection, data sharing, and data usage is paramount. Because if we collect data and it's not able to be used by biopharma, and vice versa, if biopharma collects data, I'm going to put the stake in this ground. If biopharma collects data on rare disease patients or any patients at all, and it's not available back to the patient communities, and we're doing a disservice to both sides. So it's really pushing that forward. So happy to answer any questions at the end, but I'm really, really happy. I'm really, really proud to be able to turn it over to our other panelist. And I'm trying to advance my slide.
No problem. I'll just take a moment to introduce Michael to Vanessa. Thank you so much for sharing that. I personally have learned so much from you in the time that we've worked together. So thanks for that amazing presentation. So as Vanessa said, we're going to turn it over to Dr. Michael Grosso, who is the head of clinical development and specialty medicine at Daiichi Sankyo. And as global head of clinical development, specialty medicine, Michael's responsible for preclinical, clinical, phase 1 through 4, and the life cycle management of specialty medicine portfolio globally for, and get ready for this, orphan rare genetic disease, immunology, pain, neurology, inflammation, ophthalmology, cardiovascular, metabolic, and thrombosis, thrombolysis, hematosis, therapeutic areas. He is a board-certified academic cardiothoracic surgeon with 14 years of experience in the pharmaceutical industry. And Michael, I don't know how you have time to get all that done, but we're excited to hear from you.
I delegate a lot of work, but thank you, Carrie. Thank you, Vanessa. Thank you to Global Genes for the opportunity to participate in the Rare Drug Development Symposium. I've attended in the past, and this is my first time for the opportunity to present, and I really appreciate it and respect the great work you all have done. As Carrie mentioned, next slide, please, Vanessa. I work for Daiichi Sankyo. I've been there about 11 years now. It's a mid-sized pharmaceutical that started 100 years ago or so in Japan and merged into a global pharmaceutical back around 2007. But traditionally, the company in all those years was focused on cardiovascular metabolic disease, things like hypertension, diabetes, and atherosclerosis. But the leadership of the company was smart enough to recognize that with the growing and aging global population, there was going to be huge unmet medical needs in other areas. And they undertook lots of different research and surveys and recognized that the real unmet medical needs going forward would be in the areas of neurological diseases, cancer oncology, and in rare diseases. Next slide. And so with that, we made a switch from focusing on developing traditional oral small molecules to treat very widespread diseases like, as I mentioned, hypertension, diabetes, atherosclerosis, and recognize that to conquer diseases like cancer and rare diseases, we were going to have to make a big commitment to understanding new therapeutic modalities, things like antibodies and antibody drug conjugates, nucleic acid therapy, gene therapy, cell therapy, new drug delivery systems and vaccines and so forth. And with that, the company did make that commitment and focused our research laboratories many years ago to start this work. Next slide. And with that, the programs has finally started to mature to the point where we now have 3 programs in clinical trials treating patients to test our potential therapies in 3 rare diseases: Duchenne muscular dystrophy, pseudoxanthoma elasticum, and an ultra-rare disease, fibrodysplasia ossificans progressiva. But we also have programs that are just about ready to start in patients in other rare diseases like Netherton syndrome, glycogen storage disease, chronic inflammatory polyneuropathy, rare eye diseases, including retinitis pigmentosa, and several others. Next slide. But we also recognize that there were special strategies and tactics that would be important to establish a foundation for success if we were going to be able to bring treatments to patients with rare diseases. And our goal again was to develop world-class specialty and rare disease treatments and establish a clinical development capability for our what we call specialty medicine assets, utilizing these new technologies and modalities that I mentioned to you. We also recognize that we need to be able to quickly determine which one of these therapies have the most promise, and therefore we've put in a lot of mechanisms in our preclinical work to quickly demonstrate whether or not these molecules are actually working through mechanisms that we call proof of the mechanism or proof of concept. But we also recognize that there were important tactics that needed to be put in place that were different from traditional drug development. And first and foremost, we needed to recognize that we needed to fully leverage our strength in science, medicinal chemistry, manufacturing, clinical trial design, and execution. And I'm proud to say I've been with the company now for 11 years because of their, what I believe is their great commitment to science. We also recognize that we have knowledge gaps, that we're entering fields that we don't have experience in or have little expertise in. And with that, we need to either in-license or partner or collaborate to accelerate these technology gaps. And I'll share some examples of that in an upcoming slide. We also recognize that we need to interact quickly with the academic communities, the key opinion leaders, the key experts, the academic research organizations, in order to obtain natural history studies or access to registry data, and not through just these groups, but through groups, as Vanessa has pointed out, like RareX and other foundations, in order to be able to, again, rapidly gain as much knowledge as we can and be able to move these clinical development programs forward. Finally, we recognize the importance of working not just closely, but as real partners with the patient advocacy groups. And again, I'll give a couple of examples of that. And finally, utilize and leverage the expedited regulatory pathways that may exist for rare diseases. And what we found is that our foundation partners and patient advocacy groups know this better than we do. And again, having them as a partner is a fantastic benefit and experience. Next slide. So I did mention that one of the key foundations for success is recognizing where the knowledge and technology gaps are, and we certainly have done that. And with that, in order to bridge those gaps and accelerate the learnings, we've been very aggressive about forming collaborations with companies such as ERS Genomics in order to get our CRISPR gene editing therapies quickly up to the latest developments, and also companies like Ultragenyx, who have great experience and have already launched treatments in genetic diseases. Next slide. And as I mentioned, one of the most important foundations for success is not just collaborating, but truly partnering with the patient advocacy groups and foundations that represent these patients. First and foremost, they can be the primary source of clinical information on the disease, particularly patient databases and natural history where the medical literature itself may not have lots of information. They can communicate and negotiate with regulatory agencies, sometimes to even a greater degree than the pharmaceutical industries can. They've been involved with clinical trials, and they can help us design clinical trials and avoid all the some of the mistakes that have been made in the past. And most importantly, identify clinically meaningful endpoints, things that really will make a difference in the patients' lives that they represent. They also obviously help connect us with the patients and enhance enrollment for the clinical trials and spread the awareness of the diseases. And we've been fortunate so far to work with 3 great patient advocacy groups, Parent Project Muscular Dystrophy for our DMD programs, PXE International for our pseudoxanthoma program, and IFOPA for the rare disease fibrodysplasia ossificans progressiva. And I can tell you, I've been, as Carrie pointed out, I've been in the industry about 14 years now, and one of the best experiences I've had, one of the most rewarding experiences I've had, one of the most collaborative experiences I've had has been working with these patient advocacy groups. And it's just been a tremendous experience working with these groups, and we're really looking forward to working with others as we move the rest of our research pipeline forward. Next slide. We're also trying to hold ourselves to a very high standard. We don't just want to walk around and pat ourselves on the back. And so we've put into place some very objective measures about how to determine whether we're succeeding or not. And we broke those down into 3 categories. First and foremost, the number of rare disease designations we get from regulatory agencies, things like breakthrough therapy, orphan disease designation, and in Japan, Sakigake designation. We're also going to measure the actual number of clinical trials that we really have underway. I mentioned we have 3 already. And finally, the number of new therapies we actually bring to patients. And I'm glad to say, even though we just started this venture just a few years ago, we already have 2 compounds that have received regulatory designation as either an orphan drug or Sakigake. And we hope to have another 2 programs underway in this fiscal year. So by the end of our fiscal year 2021, which would be March of 2022, we hope to have at least 5 clinical programs undergoing in 5 different orphan and rare diseases. And so with that, I'm happy to share some of the challenges that we faced in making this pivot from a traditional cardiovascular medicine company to a company that now is focused on orphan and rare diseases. But I can tell you would not have been possible without all of the help and collaboration and partnership from the foundations and patient advocacy groups that represent these patients. And so I'm happy to be part of this discussion today. Thank you.
Thank you so much, Michael, and thank you, Vanessa. We're getting some questions from the audience, but I thought that I would start with one that I think both of you might be able to answer, which is, what are some of the most common pitfalls or roadblocks that you see come up in rare disease research? Who wants to take that one first?
I mean, from my perspective, it's recognizing that the traditional ways of doing things are not going to work. The standard drug development processes that have been so successful for the pharma industry in bringing new drugs to the market and to patients just don't hold true for rare diseases, in the sense that, number one, in many of these diseases, there's very limited numbers of patients. And so you can't do the trials that are designed as the way in the past, where it took thousands of patients whom you had access to in order to get a statistically relevant endpoint to prove that the drug or the therapy that you're testing actually work, with clinical endpoints like how many patients actually, or how much longer they live. or changes in function of the heart and the lungs over time. What we've learned is in order to bring these drugs forward in the development with the very limited number of patients that are available to test it in, we have to use very different techniques and strategies. And we're starting to focus on a lot of different ways to measure success, looking at changes in biochemical markers or changes in imaging parameters, things that traditionally the regulatory agencies did kind of frowned upon as true indicators of actual treatment success. And that's where the patient advocacy groups, I think, and foundations have come into a great role, because they've actually educated the FDA and the other regulatory agencies to realize the benefit of these surrogate endpoints and the importance of these surrogate endpoints, and the realization that there's never going to be any treatments brought to the market and to these patients unless they start to accept these and build in some flexibility in their programs. And it was important that it didn't come from the pharmaceutical industry, because it kind of looked self-serving when we said it. But when it came from the patients and the patient advocacy groups and foundations, it made a huge impact. And I think there are numbers of examples of that already that we have seen. And I think that's one of the big differences that I've seen in working on programs that are focused on rare diseases as opposed to more traditional diseases. Vanessa?
Thank you. Yeah, I was going to say, Vanessa, from your viewpoint.
Yeah, I think that the big thing, Michael, you brought it up actually in your presentation, where how you partnered with the organizations, where you said the patient advocacy groups actually have the most up-to-date data about how the presentation of the disorder is happening. And that I think that happens with all of our groups, where we have publications out there, but they tend to always be, by the time they're published, 2 years down the road, they're defunct already. There's different things that we're seeing in the progression of the disorder, especially as the community gets older. So I think one of the biggest pitfalls that I see kind of with every group that I've ever worked with and that are coming onto RareX is when we go to collect data, everybody has the best of intentions when they start. And what ends up happening is, without kind of an eye on standardization and an eye on all the stakeholders involved, you tend to collect a fair amount of data that ends up getting a little bit noisy, a little bit messy, and not so useful for the end game. I think that all of us as rare disease patient groups, it ends up being that we want therapeutics. We also want to see what interventions work on our families and our kids. And to do that, really collecting data in a more standardized way is really important. So I think that has been the biggest pitfall. I mean, Kari, we've worked on data aggregation projects together in diseases that look very, very similar. And the way that we collect data became an issue, because we couldn't do cross-disease research because it wasn't a standardized way. So I think that's the biggest pitfall, is when you are told that rare disease groups go, okay, we're going to collect data, without kind of taking a step back and saying, okay, what data do we have available? What can we learn from it? If we have nothing, let's make sure that we start some standards and make sure stakeholders are all at the table at that first juncture.
And maybe Vanessa, I'm sorry.
I was going to say, I completely agree with Vanessa. Yep. The first iteration of our natural history database, we collected everything under the sun and felt like it was going to collapse under the weight of all of it. So you're 100%.
And more data is not always better. That's the thing. The more data is always better. Or not always better.
And I think one thing, Vanessa, that I've learned, the pharma industry has been very protective of their data that they collect, and we're never going to make the progress that needs to be made unless we're all willing to share data. And the patient advocacy groups and the foundations have held the pharma industry's feet to the fire to share that data, and I think that's been a great thing.
Yeah, it has been.
That was one of the questions in the question box, which was, how can foundations encourage biopharma to share data and specimens they collect on rare disease patients?
I mean, it may be a little bit bold, but some of the foundations or groups that we've worked with have made it an upfront statement. Unless you're willing to share your data that you collect with all of the groups that we work with all of the industry people that we work with, at least to some degree, then you can't participate with us. And again, the patient advocacy groups and the foundations really are the drivers and the power brokers here. So don't be afraid to flex that power.
It's very true. And also I often talk when I give my larger Rare X talk is that a lot of it is the technology that exists for data sharing and data aggregation that existing today and didn't exist back, when rare disease was kind of getting its start. And I always say trusting the tech and now enabling better governance towards it, where they have the flexibility to share data in a way that can make everybody comfortable. Obviously, and when you're coming into IND, when an approval's on the line, it becomes a little bit more dicey. But there are ways to do it that weren't available back in the day and weren't an option. And I think really kind of coming into this century in that sharing and that collaboration is really important. And the patient groups requesting that and saying, we know this exists. We might not have it at a thing. But we know it exists. And how can we work together to do that?
Vanessa, this is like taking a little bit of deep dive because people are saying, we know you love data. So, but what kind of data? Where do these new starting out organizations collect it? And they need some direction for new rare diseases starting a foundation. So what is the data, what kind of data, and where do they collect it?
Okay, let's start first, but I'll address the foundation thing. But when your community is getting together, oftentimes we have amazing like Global Genes and NORD and all the other kind of helpers and to create foundations and organizations. At the end of the day, that takes time too, right? To create an organization, to create a foundation, to create a nonprofit, that is no longer a pitfall to inhibiting data collection. You can start data collection prior to having a community or an official organization. So we'll talk about the establishment of a foundation in a little bit. But in terms of the data collection aspect of it, there's a lot of efforts out there, including RareX, where it is that anybody can come onto the platform, even if they don't have an organization, even if they might be on their diagnostic odyssey, right? So they're not even sure what their diagnosis is, but they know they're in the rare categories. They're still going through their genotype, phenotype sort of thing, but they're wanting to say, hey, I'm here. I've got something I know is rare. I don't know what it is yet. I don't know where it's going to go, but I want to at least stand up and I want to be counted. So there's efforts like that through RareX. If you're a foundation that's interested in starting to collect data and working with biopharma partners, making sure to reach out to efforts that are out there and evaluating what you need versus oftentimes patient groups, they go into what they want and it's really, really big. And it almost is impossible to get your arms around, especially as you're starting. It's backing it up to say, what do you need? Can you govern it yourself? And then how do you put it into place? And also, can you budget it? Because it's not always cheap. I know Kari, D15Q, we spend a lot of money on our data collection efforts, but I think that that's the biggest thing, that taking a step back and identifying what you need, engaging your key opinion leaders in the space, whether it be academics, whether it be clinicians, the people that are kind of rising to the top. Rodney was just on the last panel. He's one of those in a lot of the rare disease space, especially in neuro, that he's a great key opinion leader to reach out to because they can network and find the people that can help you collect, set up your data structure. And additionally, always knowing that bringing it back to the patients, meeting their needs too. Meaning you're asking the questions that make sense to them and that's going to engage them in that data collection. And also letting them know when your data is being used. What are you going to do with it afterwards? How are you going to tell them how many people enrolled? What are the symptoms that are coming out? It doesn't need to be big papers. It doesn't need to go into the biggest journal that ever lived. It just needs to get out there in some space and get back to the people like Michael, where the biopharma partners are at least saying, hey, that phenotype looks really similar to this phenotype, which is in our pipeline now. So maybe this is a good conversation to start. And I think that's kind of when you look at that holistic view is, more data is not always better. That's the first case. Any question you ask should be able to be followed up later, but you don't need to ask all the follow-up questions from the beginning.
Excellent, Vanessa. And in the 2 minutes we have left, so not a lot of time, Michael, there was a question following up to Vanessa's. You have great partnerships with patient advocacy groups, but how should a new foundation seek out an organization such as yours to drive interest? As a follow-up to Vanessa's last statement.
That's a great question. There's probably really no formal process for that to happen. But I would say don't hesitate to pick up the phone and start to reach out to any name that you can find on the website on a particular pharma that seems to be interested in any area that's even remotely close to what you're interested in. You'll probably have to, reach out to a half a dozen people before you finally do reach someone who is either willing to talk to you or knows what you're talking about. But I think persistence, I think Vanessa and Carrie, you can point to it, the success of the patient advocacy groups and foundations I've worked with all comes down to one thing, just persistence.
Excellent. Thank you both so much for being on today's panel and sharing such amazing information. I can't wait to knock on your door, Michael, and Vanessa can't wait to collaborate some more.
Yep.
I'd be happy. I'm looking forward to it.
Rodney, I'll turn it back to you for closing remarks.
Great. Thank you so much, Michael and Vanessa, for leading that great discussion. And Kari, I mean, this was just amazing. I was sitting here having my lunch and just taking notes furiously. Rare X, I think that is the bomb in the vernacular. And Michael, cannot wait to collaborate with you as well. So thank you to the audience for your questions. I would like to also thank Global Genes and co-host the Orphan Disease Center, Penn Medicine, for helping make this happen. So I would like to say for closing remarks, in addition to thanking all the speakers of our panel and discussion leaders for today, would like to thank our symposium sponsors, including Greenwich Biosciences, Horizon, Pfizer, Applied Therapeutics, Catalyst, Daiichi Sankyo, Harmony, Ionis, Janssen and Johnson, Passage Bio, Farming, Regeneron, Spark, Tisha, Ultragenics, Amicus, Avidity, Check Rare, Forge, Genentech, Ovid, and Regenix Bio. Thank you all for your support. And next, please join us for the upcoming office hours. Next here for one of the two office hours, as we've been talking about these office hours earlier and throughout the entire day. Join us, whether it be for the preclinical or clinical discussions, to really continue this chat about the journey, how to work with foundations, how to work with academics, how to work amongst all of these people, and how to find those connectors who will help enable you move your portfolio or move your foundation forward. So thank you. And finally, please take a few moments to respond to the polls that are being launched now. We all appreciate your feedback on the sessions and want to ensure that we are consistently advancing our programming in accordance with the needs of the community. And you could also share your feedback via events@globalgenes.org. And one last note, take some time, if you can, to explore the event page, because the next 2 days is going to be wonderful, excellent, fantastic, you name it, just great lineup of speakers and content. Visit the posters, build your itinerary for the remainder of the week, and plus visit the Achievements and Badges tab to learn more about how you could win a pass to a future Global Genes program, which would be really neat if we could be in person again one day. So with that, thank you again, and I will sign off and go into the Preclinical Foundations room. And Kari, do you have any last words?
Well, I'll be in the clinical research room, so I look forward to having you all there with us. And thanks again, everybody, for participating today. The chat has been really robust, and I hope you've made new connections, and really appreciate the thoughtful questions that people asked during our panel discussions. Thank you all so much.
Thank you.